Hi, you guys, and welcome to Circulatory still, and we are in part 4. So in this section, we're going to talk about blood pressure. It's something that everyone has had measured. You've watched it, but do you really know how it works? It's actually quite amazing. It's brilliant the way that was figured out, because you want to be able to measure the pressure inside this artery, your brachial artery, your artery in your arm, without having to drill a hole in the arm and in through the artery, correct? So this is a brilliant way of doing that. And I'll show you how it works. So you know, as in the picture, that you have a cuff put on and you inflate that cuff. And there's this artery, this brachial artery that's coming down your arm here, very big artery. And the idea is you're going to measure your blood pressure in that particular artery. So it'd be nice to be able to measure it right on the aorta as it comes out of the heart, but that's obviously not easy to do, so we're doing it in the arm. So you're going to apply pressure with a cuff and inflate the cuff and inflate it until no blood is coming through that artery, which is a little scary to think, right? That you've completely stopped the flow coming through that artery. And when the flow is completely stopped, see if this makes sense, and you would listen to it with a stethoscope, what you would hear is nothing, because there is no flow going through that particular artery. Now, if you deflate this just a tiny little bit and a little bit of that blood starts to squeeze through, then you should be able to hear that turbulence, because it's kind of like having your finger on a hose. It's going to make that noise of that turbulence. So at the point, you're watching the pressure gauge, at the point at which you first hear that turbulence, that's going to be the highest of the two pressures that you have in that artery. And why do we have two pressures? Because the heart is pumping and relaxing, eject, fill, eject, fill. So the pressure is always going to rise a little bit and drop, and rise a little bit and drop. And so you have two. You have the pressure of the rising pressure and the one after it dropped. So that first one, which is the highest of the two, you're going to hear first. And then you go releasing that pressure a little bit more, until it's going to become quite quiet again, because the turbulence is gone. And the turbulence is gone at what point? At the point in which the cuff is allowing the flow of that blood without any interference. Okay, so listed here I have, listen to the Korktopf sounds. Korktopf is the guy who figured it out. The first audible sound is systolic pressure. Systolic is the higher of the two. When the pulse is inaudible, when you can't hear anymore, that's the diastolic pressure. And then if you keep releasing, it's just going to drop more and more. Okay, so with a stethoscope and a cuff, you are able to, through listening and through measuring that pressure, know what these two pressures are. I'm going to backtrack to one slide. Look at this slide. You have millimeters of mercury in pressure, and this is your heart. It's like pump high and then low, and a high and a low, and a high and a low. So the high is the systolic, the low is the diastolic, and it's labeled here for you. Okay, now if you inflate your cuff pressure very high, above the systolic, and then you go deflating and it goes slowly, you still don't hear anything, boom, you hear something. You hear turbulence. So that is the highest of your pressures. Then you continue to drop, you're still hearing turbulence, it continues to drop, and at some point you don't hear any turbulence anymore. That then is the diastolic pressure. So this diagram should help you with that concept. This is another way of illustrating this. At first you would have the artery closed, so there is no sound because it is completely closed. There is nothing going through that. In section B, the artery is partially opened and you're hearing turbulence, and the diagram there is showing turbulence in it here. And then the artery completely open where the sound is going to die away. And that's the last section of circulatory that we wanted to talk about. Now we're going to give a little brief attention to the respiratory system, because those red blood cells coming from the circulatory are coming up to the lung to load up with oxygen. So we'll want to know how they do that. How do they exchange those gases with the environment? And here's a picture of your respiratory system with the bronchi and the trachea and the lungs. The gas exchange with the environment occurs at a respiratory surface, which, depending on the animal, is going to be really different. So if you're a worm, like the picture on the left, correct, you have enough surface area that you could actually breathe right through the skin. Because there's so much surface area, you could get many of those blood vessels in that vicinity. In the picture to the right with a grasshopper, now this is a chunkier animal, right? He's got more mass to it and not all of those. There's some cells that are deeper within it that are not in contact with the surface. So now it's got to be more internal. You've got to have some sort of opening to the outside that's going to bring that air in down into those cells. So it's a more complex system. Now, all these respiratory surfaces, I'm telling you, have some similarities. They have a large surface area, whether they're inside, completely inside your body, like the lion, or whether they're sort of superficial on a gill, like a fish. It's still an incredible surface area for you to captivate that oxygen. It needs contact with the air or water if you're removing oxygen from water. And it needs to be moist. When we look at the human respiratory system, there are a few of these vocabulary that I want you to know. So your sinuses, you know, the nose, your nasal cavity, you know, you understand the mouth, the tongue. Epiglottis, what is that? So the epiglottis is a lid here. It's this lid of cartilage that we can close over the trachea, preventing food from entering the trachea when you're eating. Because we have a shared tube, which is called the pharynx, for a little piece here we share where air comes in and where food comes in. And so we have to divert that once it comes in. So the food is going to go down the esophagus and the air is going to go into the trachea. So while you swallow, you want to be able to put a lid on the trachea and make sure that the bagel does not land inside your respiratory system. Next is this other piece of vocabulary is larynx. And the larynx is a series of cartilages that you have in this region. Like the Adam's apple was one of them. Then you have the trachea and you have the lungs. If you take a breath, think about where the air goes. Well, it comes in through the nose or the mouth. Nose, it goes to the nasal cavity. In the mouth, it goes straight into the pharynx. This is the shared term, shared for air and for food. And then it goes down the larynx. So in blue here is the larynx picture. So all these cartilages of the Adam's apple, that region that you can feel if you touch in the upper part of your trachea are part of the larynx. There are actually 9 different cartilages that make it up, but you don't need to know that, just that region. And then into the trachea. Then it goes down into smaller and smaller, we can just call it tubing, until it arrives at the respiratory surface, and those are the alveoli. So these grape-like structures there are the alveoli where air is going to cross. So here it's showing the trachea branching into, first of all, 2 bronchi where the arrows are, one leading to each lung. And then it'll branch again and again, and we use the word eoles, bronchioles. So the bronchioles are the little bronchi, so they get smaller and smaller until they end up in the grape-shaped alveoli. So you have millions of these little balls, these little alveoli that are surrounded, as you see in the picture, by capillaries. And in this enlarged picture, you see the capillary surrounding that alveolus, ready to receive that oxygen that will cross over, and ready also to pick up carbon dioxide that's coming from the blood. So you will get rid of waste and exhale out CO2 and bring in O2 into those capillaries. I had to show you this, the smoker's lung. There's a little video on it in the other section. Look how absolutely darkened that lung is, just from those little carbon particles that get trapped in there in the alveoli. And the pink one to the right. I want to tell you about how you breathe. It's interesting that we think about how one breathes. How do you get air into your lungs? Well, I breathe in. Well, that's not a good enough answer. How do you get air to enter? Right? It's like a balloon. How do you get air into the balloon without really pushing it in there? So look at the thorax of both of these. So we have a thorax like this, a big chest area, and this chest area. And so they're like a box. I think of it as a box. So if you enlarge the size of that box, then you create negative pressure inside that box and air tends to flow right in. And if you squeeze the box, then you increase the pressure in the box and air shoots out. That's how we breathe. So what you do is you bring your diaphragm, notice here it's arched, and we want it, if we bring it down flat, like this, and these ribs, if we were able to move them up a little bit, you would make that box bigger. And by making the box bigger, that's exactly what you do. You elevate your ribs a little bit. And when you do that, the box gets bigger, negative pressure is created inside the chest, and air naturally flows in. So the last thing that happens is air in. You don't bring air in and therefore your chest expands. It's the other way around. Your chest expands and then air comes in. And then your chest retracts and air goes out. So just the mere relaxation of those muscles in the diaphragm and in the rib cage allow those lungs to recoil and you expel air. So this is sort of the absence of a contraction, unless you force it. You could go and force air out, in which case you would use intercostal muscles also. So as a summary, airflow in the human respiratory system, you have a contraction of the diaphragm. Air flows into the body from the nose and the mouth into the alveoli. There's gas exchange at the alveolus. Alveoli is a plural. You have relaxation of the diaphragm and air flows out of the body. So in this summary, I'm just saying the diaphragm, it's also your muscles that are between your ribs. And if they're between your ribs, they're called intercostal. Intercostal. One last thing here in Investigating Life, where they have research at the end of your chapter. It tells us of a very interesting case where not all animals use red blood cells to transport oxygen and CO2. So there is this fish, the ice fish, that lacks red blood cells and hemoglobin. And what happens is that those respiratory gases are transported just dissolved in the plasma. They don't need hemoglobin like we have. It's very, very cold waters. So the metabolic rate of this fish is so low. It's like barely breathing and barely metabolizing. It does everything slow motion. So the lack of this mechanism to get oxygen to tissues quickly, which is what we use with hemoglobin, it's not really needed. And the reproductive success of these organisms is not affected by not having hemoglobin to transport that oxygen. So they can just do it slowly in extremely, extremely cold waters. It's an interesting story. And that was the last of your slides. And congratulations, you have reached the end. I should have this great slide at the end. I'll just dry it out. And congratulate yourselves. You have listened to many of my lectures and reached the end. Good for you. Stars here. Take care, you guys.